Hello AP Biology students. So I just wanted to give you some very last minute tips to remember before you kick that test in the teeth. So listen closely, I'll be as quick as I possibly can. Hardy-Weinberg problems. You are going to see a Hardy-Weinberg problem, and we've done a little bit of review in class, but I want to give you guys the key to success on all of these things. So the number one thing that I think you remember is you have two equations. You have P plus Q equals 1, where P is the frequency of the dominant and Q is the frequency of the recessive, but most of the time, the number one thing that you're going to need to know is that if they give you the recessive individuals, like non-tasters, that is something that is often a recessive trait, you need to set that frequency, which you find by adding things together and dividing by the total, to Q squared. Q squared is your key. That's a frequency of recessive individuals. Don't use Q. Use Q squared and then take the square root of that to get Q. That's the secret to Hardy-Weinberg problems. Chi-squared problems, you're gonna probably see these too, maybe a couple of times in different formats. Remember, if you're gonna use this formula, this chi-squared equals the sum of the observed minus the expected squared divided by the expected. And remember, the observed is what they actually get from the data. The expected is what you would do either from in animal behavior. You're usually going to set it to just the average. So it's just 5 go one way, 5 go the other way. In a genetics problem, you're going to set the expected to whatever ratio you would get from the Punnett square. They could use sex linkage, where you have to set up the X and Y and the X and the X. They could use regular or a dihybrid cross, where it's 9 to 3 to 3 to 1. Just remember, if the chi-square is high, reject the null hypothesis. There's probably some offensive thing that you know. If the P is low, well, if the chi-square is high, reject the null hypothesis, or HO. For animal behavior, remember, the null hypothesis are just differences are due to chance. Just 5 went one way, 5 went the other way. They don't prefer one side or the other. With genetics, it's the null hypothesis is that the breeding does follow predicted ratios. So if it's very close to the 9 to 3 to 3 to 1 ratio, if it is close to 75, 25 in a traditional just heterozygous, double cross. Rejecting a genetics problem is a little tricky because it probably means that there's some different type of inheritance. It isn't the one that was suggested. So that could be because it's on a sex chromosome, or it could be linkage. Remember, we studied linkage in the fly phenotypes, and if it isn't 1 to 1 to 1 to 1 with the linkage, that's probably going to mean that you can reject the null hypothesis and there is linkage. It means they are on the same chromosome. Regulation, we talked a lot about regulation. Most regulation is due to transcription factors. Remember, we talked about that with cell signaling. You get something to come to bind a receptor, that's going to cause an amplification, and then that's going to make transcription factors, which are going to turn other things on. That's how you get the cell signaling pathway. This is a type of transcription factor, just something that causes transcription to either occur. If it's an activator or repressor, it won't occur. But also there's interfering RNA that can come and cause the mRNA to be broken down. And then also, remember, just regulation, P53, it comes up often, is a tumor suppressor. And if it doesn't work, if it mutates, then you're going to get cancer. And along the same lines, CDKs, cyclins, if you hear about CDKs or cyclins, they check the cell cycle. If they're not working, then you might get cancer, you might get uncontrolled cell growth or division. Remember these things. Homeostasis, just think about homeostasis. That's returning to normal. That's if you have too much of something, it needs to return to a normal set point. And at a molecular level, that's where if you make something and that activates an enzyme, and that activates another enzyme, which makes something, which activates another enzyme. In that process, in that signaling, the last product is probably going to block a step earlier. That way you don't continue to make things. You get enough of it, it's going to turn off things upstream so that enzyme stops making things. It can also refer to like thermoregulation, where if we're too hot, we sweat and our blood vessels dilate. If we're cold, our blood vessels constrict and we shiver. It keeps us at a normal 37 degrees Celsius or 98 degrees Fahrenheit. And hormones are usually involved in return to homeostasis. Remember, endocrine signaling, you're going to make something that's going to go to another cell, some other tissue far away. So that endocrine, it's like insulin. We didn't talk too much about it, but insulin will lower. It's a hormone in the kidney. You're going to lower, keep water regulation in a tight balance. This is all homeostasis. Thinking about membranes, you need to know why membranes exist. They're kind of barriers to make sure that you keep the right stuff in, the wrong stuff out. Small nonpolar molecules, these are like oxygen or CO2, they are nonpolar, or other things, they can go right through the membrane. Something else that goes through the membrane but requires a channel is water, remember. That's the whole osmoregulation. Water can go through aquaporins. But other things, big things, or polar things like epinephrine need to bind to a receptor. When they bind to a receptor, that's going to cause something else to occur. It can cause a secondary messenger like CAMP to be used, but it's going to cause some process to occur and some response to occur downstream. Membranes, remember, nonpolar can get through, polar has to use some other mechanism, some receptor to cause its function or its effect. Cell respiration. You don't have to memorize every single step, but it's good to know what the most important steps are. And remember, it's occurring in the mitochondria. And the most important part of cellular respiration, where you get the most ATP for your buck, is the electron transport chain and oxidative phosphorylation. And specifically chemiosmosis, remember, this is where you form this gradient, these electrons coming through. You're going to form this gradient of hydrogen ions, and when they go through, they make ATP through ATP synthase, probably the most famous enzyme. And remember, NADH is used in cell respiration. NADH, which is created, it's reduced, remember, it's reduced to make NADH, and then later oxidized to make NAD+. And if you're trying to remember oxidation versus reduction, which one's which, remember Leo Ger. Leo says Ger. Leo says Ger. Loses electrons, it's oxidation. Gains electrons, it's reduction. So this is cell respiration, NADH, it's got that extra H, it's electron rich, so it has been reduced. It's oxidized here though. Reduced to make it, oxidized here. That's the electron transport chain. And then quick stuff to know for photosynthesis. Remember, you have light-dependent reactions and then light-independent reactions, or the Calvin cycle. Light-dependent reactions, light comes in, they occur in the thylakoid. Remember, thylakoid, like thylakoids, a stack of them is like granum, like a silo where you store grain. H2O goes in, oxygen comes out. You're making ATP and NADPH. Remember, P for photosynthesis, so you don't get them confused. They go into the stroma, which is just the space. You can take a stroll through the stroma, that's the space. And that is going to take the CO2, remember plants need the CO2, and it's going to make the G3P, you need two of which to make glucose for them to store it. But also remember that plants do also do cellular respiration. They don't just do photosynthesis. So they will do the process in the last slide, but they also do photosynthesis. And remember, because of this photosynthesis, and actually chemosynthesis as well, this is where we get energy for the sun, so we need lots and lots of photosynthesis in order to support things later or higher up in a trophic pyramid. So there's going to be less things at the top of the pyramid eating things below them. Remember our pyramid. And producers are going to make up the bottom of the pyramid. Consumers, primary, secondary, tertiary consumers will be at the top, and there's going to be far fewer of them because of that 10% rule. Only 10% of the energy is available to higher levels to make new biomass. So remember, they lose 90%, they keep 10% as you go up the pyramid. So students, I want you to know that I am already proud of you. You guys were the very first class to do the transformation right on your very first try. You've done experiments with roly-polies, you've learned about bees, you've learned about a bunch of things, done experiments with enzymes, done experiments with photosynthesis. You have accomplished a lot in this class, and I want you to remember that. Trust the training. And also, I just want to say, Caden, Macy, Tom, Justin, Claire, Dan, Wes, Rhiannon, Allie, Ben, Ashley, Amelia, Maggie, Hallie, Caitlin, Sharice, Sydney, Tanner, Connor, Jasper, Bridget, Carrie, Liam, Bosov, Natalia, Emily, Jake, and Anita. I'm rooting for you. You should be proud of yourself. Now go kill it.